Everyone for joining this session of workshop, title of which is Voyager, Exploratory Spatial Data Analysis, Geospatial, Spatial, and Lambda Moses. I'm just conducting our workshop. I don't think we may not have a moderator, but I will keep my eye on the Q&A. And so, thank you and welcome.
So Bioconductor workshop, it's hosted on workshop.bioconductor.org. It's on Galaxy. So yes, if you go to the workshop Galaxy, you on the left scroll down and navigate to the Voyager Exploratory Spatial Analysis. Click launch. Yeah, it might take a while.
Come on, Alex. You suck stuff. I was probably once I did.
If you're interested, you can click visual. This outline for content to navigate. All right, so while you're setting up, I'll do the intro. So this website has the rendered version of this workshop. So here I expect you to be already familiar with SingleCellExperiment. And that's a breakdown. So first I will do an intro to the Voyager project, and after that we will we'll learn to use the SpatialFeatureExperiment object, and then we'll perform some exploratory spatial data analysis on a real data set.
They changed the room.
Yeah, so if you want to take a look at the workshop, that's just a rendered version. So let me get to the slides. Hi, so hi, my name is Lambda, and I'm a postdoc at Lior Pachter lab at Caltech. Yeah, so this is about the Voyager project, which brings the glorious tradition of geospatial data analysis to spatial omics. We already have a preprint, and if you want to read more about it, you can scan the QR code. That's the first version, and I'm mostly done with the second version. Yeah, I think because we already have a lot of spatial contents here, if you're here, I think you already know why we should do spatial, so I skip that. Yeah, also, okay, so if the number of new publications over time can indicate interest in given field, then we
So I'm sorry to interrupt, but the people online can't hear.
Oh, sorry, let me see.
So we're just going to figure out what's going on here.
I think close to the microphone. Microphone is too far.
Is it?
I think there's some unmuted.
Oh, is it mute?
No, that's the video.
Audio.
Is anybody on the session right now? I see Alex jumping on here. My son.
Yeah. Oh, right. Like a touch screen isn't like comprehensive.
Okay.
Does it work?
It seems so.
So, some people may have actually been giving moderation permissions. So, all the people who are remote, please make sure that you're muted, just in case. And it's just music. All right, you should. Sorry about that.
Sorry. Okay, so for the people on the line, yeah, you can scan the QR code to, if you want to read more about the preprint. Yeah, the slides are on the GitHub repo for Voyager workshop, which is linked in the workshop. So yeah, I'm now confused. So, yeah, so if the number of new publications over time can indicate the interest in this field, then from this plot we can see that spatial transcriptomics has drastically grown since 2019. Also, like from how the amount of spatial content in this conference this year and how how much it has grown since last year, I can tell that spatial transcriptomics has come of age. So from this plot, about 2019 is where it has really taken off. However, spatial data analysis has existed for decades before the rise of spatial transcriptomics, mostly in geography. This is a timeline of some of the classic publications for some of the methods used in Voyager, and we see it dates back all the way to although this field is still continuously evolving. There are many different kinds of geospatial data, and for each of them, there are many well-established data analysis tools and analysis methods and software implementations. So if we can draw an analogy between certain types of spatial omics, well, here I focused on transcriptomics to geospatial data, then we can borrow some of the existing tools instead of reinventing the wheel. So far there are actually already over 130 different spatial transcriptomics technologies, and there are even more for proteomics and spatial epigenomics and so on. But however, only a handful are very commonly used. So here, this plot shows the number of institutions using each spatial transcriptomics technology, and we see here by far Visium is the most popular and it's used by almost 200 institutions around the world. So because of the popularity in this workshop, we will use a Visium dataset. Although, like in the on the Voyager documentation website, we have tutorials from many other technologies like Merfish as well. So in case you don't already know, this is how Visium works. You have a microscope slide, and you have here you have the areas to the tissue capture areas where you will mount the tissues. In each of these areas, there are 4,992. Well, basically like almost 5,000 spots, and these spots are in hexagonal array. So in each of the spots are printed so each of the spots is has printed oligonucleotides, which has a barcode whose spatial locations are known, and you have UMI and the capture sequence like a poly-T to capture polyadenylated mRNA molecules. And so here after you mount the tissue on this array, and after you permeabilize the tissue, these oligos will capture the transcripts, which are then reverse transcribed and amplified and sequenced. This array is so each spot is 55 micron in diameter. Actually, in practice, they're probably a bit bigger or closer together, and the spots are 100 microns center to center. In contrast, single cells are usually, well, actually it depends on the cell type, but for many cell types, they're like about 10 microns across. So Visium does not have single resolution. So that has a lot of implications on the downstream analyses. So here, this is a schematic of the Voyager project, and here to start, I know that Voyager is based on the new S4 class called SpatialFeatureExperiment, which extends SingleCellExperiment and SpatialExperiment already on Bioconductor with existing tools in the geospatial field, such as SF for the vector geometries and Terra for the raster images. Actually right now, both Voyager and SpatialFeatureExperiment are already on Bioconductor, and they're available in Bioconductor 3.16 and higher. So now we'll get to the SpatialFeatureExperiment or SFE class. The heart of how SFE extends SCE and SPE is simple features, which is the standard way to represent geometries in the geospatial field. And in R, simple features are accessed from the SF package, which is part of the tidyverse. So it's actually very user friendly. So the spatial data, they're basically data frames, and you have a special column for the geometries, and the other columns are for the attributes of those geometries. But behind the scenes, we actually have a Geo C++ library for geometric operations, which can include so you can find geometric predicates like whether two geometries intersect. You can also find the actual intersections. So how are the geometries relevant to biology? So here, the plots below are taken from the example data set. So here we see these circles. So these are the Visium spot polygons plotted as their actual size relative to the cells, and these red orangish polygons are the myofiber segmentations. And so we see here from geometric operations, we can see which myofiber intersects with which Visium spot, and these myofibers are colored by their area. So we can this way, by looking at the intersections, we can relate the attributes of the myofibers to those Visium spots, and hence we can relate like an area or like a morphological matrix to like gene expression or gene expression QC metrics and so on. Yeah, also like because we SFE puts the geometry front and center when plotting. So the actual cell morphologies as polygons are plotted by default instead of the points in most of the other packages like Surat. This is a schematic of the SpatialFeatureExperiment object. So in the center, in the gray box here is SpatialExperiment. Which extends SingleCellExperiment. So here we have the assays field, which has the gene count matrices. Like you have raw counts or log normalized counts, and each row is a gene, and each column is a cell or a Visium spot. And then in SDE we have the row and column metadata for corresponding to the dimensions of the gene count matrix, and we also have reduced dims, which stores the cell or like mostly cell embeddings in lower dimensions like PCA. So how SpatialExperiment extends SingleCellExperiment is that SPE adds a spatial coordinates field for the centroid coordinates for like a Visium spots for cells, and it also adds the image data field, which stores the images like H&E or fluorescent images, which can be used for visualization. So SpatialFeatureExperiment extends SpatialExperiment with the geometries that correspond to the columns and rows of the gene count matrix. So for call geometries, so these are geometries that correspond to the columns of the gene count matrix. So they can be in the Visium dataset, they can be the Visium spot polygons, or in like a single cell resolution Murphy dataset, they can be nuclear cell segmentations, and you can have like any number of geometries for these. So for instance, you can have cell segmentations and the nuclear segmentations, or cell segmentations from different algorithms, or like whatever annotations or so on. Because many spatial analysis methods will require spatial neighborhood graph, which is a graph that indicates who's the neighbor of whom and how strong the neighborhood associations are. So analogous to the call and row geometries, we have call graphs and row graphs, and so again these are analogous to the call data and row data. In addition, there are geometries that do indeed belong to this data set, but they do not directly correspond to the dimensions of the Jinkou matrix. So these I call annotation geometries or annotate geometries. So for example, in the Visium dataset, these can be cell segmentations, but these can also be like tissue boundaries or pathologist annotated histological regions from other software like ImageJ and QPath. So similarly, we have annotate graphs to store the spatial neighborhood graphs for these geometries, so we can perform spatial analyses on their attributes. So what Voyager does is that Voyager aims to be like Voyager to SFE is just like Skater to SCE. So Skater does plotting and some non-spatial exploratory data analysis to SCE objects, and also like a Seurat to Seurat object. So what is EDA? Well, in John Tukey's word in his monumental book Exploratory Data Analysis, which spearheaded this field, EDA can be summarized as the spirit of EDA is it is important to understand what you can do before you learn to measure how well you have seen to done it. However, Tukey did not talk about the spatial aspect of the data at all. So exploratory spatial data analysis, or ESDA, means EDA explicitly for the spatial aspects of the data, and especially the spatial autocorrelation. So yeah, actually, according to the R for Data Science book, which has a chapter on EDA, EDA is, you can also think of it as like a process, and which you try to investigate all the questions that come to you, and Voyager is meant to facilitate this process when it comes to the spatial aspects of the data. So what is spatial autocorrelation? So the spirit of spatial autocorrelation can be summarized in Tobler's first law of geography, which says everything's related to everything else, but near things are more related than distant things. So basically, in other words, nearby things are not independent of each other. We have positive spatial autocorrelation, which means nearby things tend to be more similar. So these are pretty common. An example is weather. So for example, weather in LA and say Santa Barbara or tend to be more similar than weather in LA and say San Francisco, Seattle, or Boston. But there's also negative spatial autocorrelation in which nearby things tend to be more different. Actually, there are examples, and if you there is an example in the Murphish dataset on the Voyager documentation website. But a very obvious example is the chessboard, in which the neighbors must be different. I'm definitely not the first person to who brings some elements from the geospatial tradition to spatial transcriptomics. So this slide lists many existing packages that do that. It also shows which aspect of the tradition is brought into spatial transcriptomics. However, these packages really tend to focus on how well you have seen to done it, and they they do very specific tasks. In contrast, Voyager tends to focus on the because these existing packages tend to focus on how well you have seen to done it. The what you can do part is unaddressed, and that's what Voyager aims to do. Because it implements many different spatial like ESDA methods for you to better investigate every idea that comes to you. Voyager has a systematic uniform interface to unite all those different methods for it to be easy to learn. And for some of those methods, I have written more efficient implementations for them to scale to larger number of cells. So finally, we are coming back to this schematic. So here in the middle shows what that uniform user interface looks like for these a large variety of geospatial data analysis methods. Because visualization is very important to EDA in general, and Voyager not only implements the not only implements the plotting functions to translate gene expression and cell attributes on the geometries with geom SF, but also implements plotting functions to visualize the results from these spatial analysis methods. We have written a comprehensive documentation website, which you can look at the website by scanning this QR code. And so we have written tutorials from many popular commercial spatial transcriptomics technologies, but we also have a codex vignette that's proteomics. And these example data sets shown in the vignettes are available in the SFE data package, which is also on Bioconductor. To make sure that the documentation and the vignettes are reproducible and scalable, we build the entire website on GitHub Actions with package down. So that's with on the fresh machine from scratch. And because GitHub Actions has very limited computational resource, so I will have to make sure that the methods are at least like somewhat scalable. And actually, I did build the vignettes on GitHub Action with like a Merfish dataset with like a 400,000 cells, and it works. And we also have the GitHub Actions automatically generates Google Colab notebooks, which are linked on the website, so you can change the parameters and explore more on the vignettes. And finally, because the single cell and the spatial transcriptomics community are kind of split between R and Python. Actually, we also have a Python implementation of some of the functionalities of the R package Voyager, and because Surat and ScanPy actually give different results for like PCA, like log fold change and so on, we wrote compatibility tests to automatically compare the output from the R and Python implementations of Voyager to make sure that they are consistent. All right, so yeah, here I can. So yeah, we can take a look at this website, and so this is a documentation website, and it shows so each technology has a landing page which gives you an intro to the technology, and and then it links to the vignettes using this technology, and then similarly for each spatial analysis method, we have say I'll pick one not shown in the workshop, and we have a landing page showing like a brief intro of how this method works, and followed by a table of the linking to the vignettes using this method. And I also have the regular like functional reference. Okay, so now let's get back to the workshop. So basically, like the first section in introduction, they're already covered by in the slides. Okay, let me make the font larger. Is that good for larger? Yeah, so first here we load the packages. Yeah, so like that's already covered in this. So I'll just jump straight to part one. So here we create an SFE object. So because Space Ranger is the official software from 10x Genomics to do like genome alignment of basic reads. So here I'll just show a toy example using like a toy Space Ranger output to create an SFE object, and that's part of the SpatialFeatureExperiment package. So yeah, here we show like two samples, and that's like the top level directory. Yeah, then the outputs that matter to us are all like in the outs directory under that sample directory, and Okay, the visual mode is, we go back to source. Yeah, so here we look inside the outs directory. And actually, like, usually there should be like way more. There, you also there's like usually there's also like a raw feature BC matrix, and there are also like HDF5 versions of the matrices. But here, for example, there's only the filtered one, which only has the. So actually, Space Ranger can also detect which it can kind of do the image segmentation, so it can determine which Visium spot is in the tissue. Although like sometimes I would still do it manually. For sometimes like Space Ranger gives like it's pretty rough. And the spatial directory has the spatial information like about the like spatial like has the spatial information like the resolution of the coordinates and so on. So here, this for example is in the MTX format. It's a sparse matrix, and in the spatial directory, we have the images of like of different resolutions. Actually, like low res is like pretty low. Like high res is still like not very high resolution. It's like only fits in like about 2000 by 2000 image-ish, but it doesn't have the full resolution. Okay, so here we use the read 10x Visium SFE function to load this into R. Okay, any questions? No. Okay. Oh yes.
Just one. Is the read 10x data? Is that function from 10x or is that from?
From me.
Okay.
Actually, for it's in more detail. So for SingleCellExperiment, there's like a read 10x function from the droplet utils package, and then in SpatialExperiment, there's like a read 10x Visium function, which is based on the droplet util function, and my function is based on the SpatialExperiment function. All right, so next, so after you read into R, well, that's just a tiny, that's like a tiny example, but here, like when it comes to like geometrical operations, we can look at like a small subset of a real data set, which may sound like more relevant. And here the data comes from the SFE data package, and on line 144, when you run the code, just note that just make sure to open the R console for it's asking you in the R console whether to create a directory to download that. Yes. Yeah, then it should be load into R. So first, let's look at the getters and setters of the geometries, and so first call geometries. Actually, there is also row geometries. It's implemented. The function should work, but like row geometries might be used for like transcript spots, like like in SM-Fish-based data. But however, like often, like in Murphish data sets from Visium website, like often those are so large, like 30 gigabytes, that like I'm not entirely sure what to do with this. So like as a result, I haven't really played around with it. But it's there. But here, like because call geometries are more often used, I'll do call geometries here. So if you're already familiar with SingleCellExperiment, I really try to emulate the conventions of SingleCellExperiment. So the user interface of call geometry emulates that of reduced dims. So reduced dims, you will like write reduced dim. And then like you put a SingleCellExperiment object, and then you put a name of the image reduction, like PCA and so on. So that's familiar. So then like call geometry is like the same. You put like the single SFE object and you put a name of the geometry. So if you don't put a name, it will by default pull the first one. And so here we use the getter, called geometry getter to get one of the geometries. So that's the Visium spot polygon. And so here you get an SF data frame, and that's what it looks like. It has the special geometry column, and it has like another column called sample ID. So I plotted. This is what it looks like. There's also a setter for called geometry, so it's pretty much the same. It's also designed to emulate reduced dims setter in SingleCellExperiment. So this is how you will you'll set a geometry in SFE. So because the getter and setter will ask you for a name, you can, in case you don't already know a name, or if you wonder if the name has already been taken, you can use the called geometry names to look at which names are currently used. But so this is also to emulate in SingleCellExperiment the reduced dims names function. Also, for certain geometries, certain types of geometries are more commonly used, such as say self-segmentation and the Visium spot polygon. They're like shorthand functions, so you don't have to type in the name all the time. So like there's spot poly and a getter and setter, like which is equivalent to what the code ran above. Okay, so that's the called geometry, and then like row geometries are like they're all the same. So for analog geometries, actually the user interface are it's also all the same. So so again, like you have the getter analog geometry and setter analog geometry, and you put the SFE object and the name of the geometry of interest. So here I'm getting the teacher boundary. Got questions or no? Okay, so if no questions, I'll continue. Yeah, so here I get the teacher boundary. Again, it's an SF data frame with geometry and another attribute. And so this is what it looks like. It's just a small subset of that data set that I've cropped. And the setter is actually, yeah. And also, just like in called geometries, we have there's a function to check the names of the annotation geometries. Like actually, in this example data set, there are quite a few annotation geometries, which includes the tissue boundaries, like the higher resolution myofiber segmentation, also simplified myofiber segmentation. We also have nuclei segmentation. Yeah, also just like for the called geometry, there may be certain annotation geometries are maybe more commonly used, like tissue boundaries. So these also have shorthands, like there's a tissue boundary getter and setter. So yeah, so that's for the getters and setters of the geometries, and next we can go to spatial graphs. Again, the user interface for spatial graphs would emulate that of reduced dims and the geometries. So the SFE package implements many methods to find the spatial neighborhood graph, and so there's a one function rule all. It's called find spatial neighbors, and it covers methods like triangulation or like different methods to prune the triangulation. And there's also k-nearest neighbor graph and distance-based graph and so on. And there are also different ways to find the edge weights, like there are distance-based edge weights, and there are also different ways to like to row normalize. Yeah, row normalization is the most commonly used way to set the edge weights. But here we do a triangulation. Yeah, that's for the SFE object, and that's for the Visium spots. And here we plot this graph here. Yes.
What happens when you put more than one
image you're
There can be any number of images.
so where would you tell it?
Oh, so this one, yeah. So actually, there are defaults. So there's like an argument here for which. Yeah, there's an argument here for like telling you like which geometry to use, and the margin argument tells it if you're using like call geometry or row geometry or annotation geometry.
Yeah. And then with like type tells you what column in column data would be, or like
A call geometry. You put the name of the call geometry.
so the column geometry is image.
Well, SF data frames.
So you have one SF data frame per image. Is that how that works?
No, they're not associated with images. They're independent from the image data field. Yeah, let me go back to the schematic in the slides. So here just now in the workshop, I'm trying to find a call graph, and these are associated with. So to find these, you will pull one of the SF data frames out of the call geometries to compute this.
But the call geometry is that one row, like that one thing in the green, right?
It's actually like the green one's for the spatial geometry column, and the yellows for the other attributes. But here to make the graph, we will only use the geometry. So
you've only got, let's say you've only got a subset of cells in each image. Like these are cells from image 1, these are cells from image 2, these are cells from image 3.
Okay, so like different like sections, right?
Yeah.
Yeah, actually
different people, different anything.
Yeah, so actually I did not mention that for simplicity. For actually this specific example data set only has one sample, like one section, but all the getters and setter functions, so that's like very unlike single-cell experiment. So actually for all geometries, there's like a additional argument called sample ID. So that way, that's how you specify like which sample you pull. But or you can put all to get all of them.
Okay, so then when you run your graph thing,
yeah. So there's
the sample, like you can't set sample ID.
You can. You can set sample ID. You can set like which one or like all. So if it's all, then the same graph finding algorithm will be applied to all the samples.
Okay. Thank you.
Yeah, so here that's this plot shows the triangulation graph. As we see here, it's not always like what you expect from the Visium from the Visium spot adjacency graph.
I'll ask the same question. So when you say plot, is it automatically just plot first?
Yeah, like actually for this one, I didn't even use like a Voyager functions from SPDEP. I just specified because the output of this function is the graph itself.
But what if you had multiple graphs for each sample?
So it's just like it's like the same thing for I'm just about to get to the getters and setters for call graphs, and here it's the user interface is the same as that of like call geometry and like reduced dim. So again, like you put SFE object, and then you put the name of the graph, and then there's like an additional argument called sample ID. So that's how you specify which sample.
Okay, thank you.
Yeah, welcome. Yeah, for Visium specifically, because it's in the hexagonal array. So and as we see just now, like those algorithms don't necessarily find the actual adjacency graph. So there's a special function like find Visium graph just to do that. And again, here we use the setter to say like Visium graph. So here when we plot it, this is what it looks like, and that's what it's supposed to be like. Yeah, and again, just like reduced dims and the call geometry, there's the call graph names to see like which graphs are present. Yeah, so like in this workshop only uses like one sample, but the vignette of the SFE package has like a 2 examples for like when you have like multiple samples. Okay, so
Just one more quick question.
yes.
The graph structure that you are working with is specific to the SP depth?
Yeah, it's from yes and no. I suppose it's called the list W. It's implemented in SPDEP, but it's not really specific to SPDEP because other packages in the geospatial tradition also use it. Like there's the ADE spatial package also uses that, and also there's the for spatial modeling, there's the spatial reg package, which also uses the list W. But however, like so initially I used list W to make it simplify that, so I don't have to do conversions. However, in like a future version of SFE, I'm considering like just using like just a sparse matrix for the adjacent graph because I found out like some of the implementations for like finding spatially lagged values are not very efficient for like not scalable enough, and if I'm going to re-implement those, if I get rid of SP depth, then I may also change the I may also like use sparse matrices, which can also be like delayed out rate and yeah, but I need to experiment with like how efficient that is.
And is there a conversion to an iGraph?
Yeah, so SPDEP also implements those functions to convert the list W to like a sparse matrix, or actually not sparse matrix, dense matrix. But I think Voyager, either Voyager or the SFE package implements a function to convert the list W to a sparse matrix. And SPDEP also has a function to convert the list W to iGraph and vice versa. Yes.
I'm just wondering about the motivation for the difference in the two networks. Like, would it be reasonable? I'm assuming that like these edges all have weights. Would it be reasonable to not consider anything that's immediately adjacent to a given spot? Like, I'm considering the edges. Like, if we look in like the bottom left corner on this one,
yeah.
You can see like the one immediately above it vertically is connected, but if you scroll down to the Visium graph, the one immediately above it is not like connected. So I'm just wondering about like the motivation for that.
So
rather than differences in weights.
So yeah, like for this one, the Visium one, like it only considers like the adjacent one, like adjacent one in like a hexagonal tessellation. While this one it does the triangulation and it just finds like a yeah, just tries to connect them all like. So there are many different kinds of graphs implemented in SPDEP, and as a result, SFE package. So actually, I haven't. It would be really interesting to benchmark to experiment like what are the effects of different graphs on the downstream results, but I haven't gotten to do it yet. But I think it's very important.
Thank you.
All right, so if you don't have questions, then we'll move on to the ESDA. The data set we are using here comes from, well, this publication so it's performed with Visium in mouse. So what they did is that over a time course, they injected the toxin called notexin into the tibialis anterior muscle, so which is the muscle in front of your calf. So that will create a severe damage to the muscle, and then over the time course, they collected the muscle to perform Visium to track wound healing. So this data set is already in the SFE data package, and as well as all the geometries. So basically, I used StarDist which is a deep learning model to perform the nucleus segmentation from the H&E. And but for the myofiber, I actually did the segmentation manually, but that was literally for a training data set for another like deep learning model. Although because I'm not an expert in deep learning, that didn't go very well. But for this time point, I did get the manual myofiber segmentations. So here, yeah, it's line 270. We load the full data set into R, and the one we used in the previous section is a small subset of this. It comes with an HD image. Okay, maybe I'll make it smaller. Just to show the H&E here. So here in H&E, we this region with many leukocyte infiltration, the blue nuclei, that's the injured site. The darker red strips are the muscle tendon junctions, and these pink areas are the myofibers. So actually, this dataset originally did not come in the standard Space Ranger format. So and because the image functionality was added in the second release of SFE, while the dataset itself was in the first release, like it's not the image does not come with the SFE object, and I'll add it here with the add image, and yeah, I'll mirror that for the coordinates to match those Visium spots. So here, let's look at what's in the call data. So it has a standard PC metrics, and but there's like in tissue, which means whether which indicates whether each Visium spot intersects the tissue. So here, for anything downstream, we'll only use the spots that do intersect tissue, and we also remove genes that are not detected anywhere. Yeah, that's just the log norm count. That just comes from scalars. That's just standard SE. In the next section, we'll perform the geometrical operations. That's even before the spatial statistics, and here we'll use geometrical operations to relate the annotation geometries to the Visium spots. And again, like here we have myofiber segmentations and nuclei segmentations. The main plotting function for call data and for gene expression in space is called plot spatial feature. So again, it uses geom_SF to do the plotting. So it plots the actual geometries. Yeah, so in this plot, you have got this plot. The blue circles are the Visium spots, and they're colored by the library size, like the total number of UMI counts, detected spots, or in counts. And plotting the background, those are the myofiber segmentations, and they're colored by their area. And just from this plot, we can see that in this area with larger myofibers, they tend to have smaller library size, while like in some these regions with smaller myofibers, they tend to have larger library size.
Is this like a ggplot object?
Yeah, it is
start
ggplot.
Overlaying stuff.
Yeah, it's ggplot, so you can like do whatever ggplotting you want.
Could I be lazy and add another boundary on
yeah.
Like is there a geometry for that?
Well, like so here when you look at the arguments here, it can plot like one call geometry and one anode geometry together. So here, like I specify the call geometry is spot poly, and then here behind it plotted like myofiber. But you can change that to like tissue boundary and so on. there's like also an argument for the image, but however by default the image is not plotted because unlike throughout, because like in my experience and in my opinion, I feel like if you plot the image behind the spots, it tends to distort the color perception of the spots. And here we've using like geometric predicates, we try to find how many myofibers intersect each Visium spot. So actually here the default predicate is intersection, but actually you can change that. You can change that into like ST is covered by or ST is disjoint and so on. Just pretty much all the predicates supported by the SF package can be used.
The myofiber was done manually.
Yes.
Is that written up in the paper about this?
Yeah, it's in the methods.
Deep learning applies, right?
Yeah, like that's sort of like initially I tried to do some manual annotations as like training data, although like when I tried to train the model, it didn't work out very well. So like there are 2 other time points, and I'm just too lazy to do all the manual annotations. So I'll talk to like a David Van Valen, who's an expert in deep learning to do that. Yeah, he's on my committee, so.
Is there an interactive tool that the annotation was done on for that?
Yeah, I used so in Fiji or ImageJ, I used a plugin called LabKit.
Okay.
So actually in Voyager itself, like because it's not, I don't really have the background, and I really stink in Shiny. So as a result, like I don't have like an interactive tool inside Voyager. Maybe yet.
I think there is one in Bioconductor.
Like IC things like that.
Yeah, someone has done one where you can.
Yeah, like I think because I think that's sort of why I kind of want to emphasize in my paper that I extend SDE and SPE, because when people have like existing implementations of like a say other spatial analyses or non-spatial or like interactive and they use SDE, then I can just use those instead of like reinventing the wheel. Yeah, so here we again we use because the number of myofibers is added to the call data, so we can plot that with plot spatial feature again. And so this plot shows the number of myofibers in each Visium spot. So because there's no one-to-one correspondence between Visium spots and myofibers, so how are we going to relate the characteristics of the myofibers to Visium spots in gene expression? So the way to do that, so again with the geometries, we'll have to use like some sort of summary functions to relate the myofiber attributes to Visium spots. Like here like by default, yeah. So we use the anode summary function, and by default it's uses the mean, but you can use like sum or median or whatever. Yeah. So here we compute the average area of the myofiber. Like an average area of myofibers that intersect each Visium spot, and add that to the call data. Yeah, so that's so that's line 355, and after that we can plot that in space again. So this shows like an area with larger myofibers. So. So again, because SFE inherits from SDE, all the other plotting function from scalar can be used. So yeah, here plot call data is just the scalar function, and that just shows the scatter plot and counting engines. And yeah, so we see here that it has 2 branches, and so for larger myofibers, they for Visium spots with larger myofibers tend to have like a smaller encounts for the same engines compared to like Visium spots with smaller myofibers. Yeah, we can do the same for like encounts and the proportion of mitochondrially encoded counts. Yeah, and that's colored by the myofiber area. So, okay, so any questions before we move to the next section? No? Okay. I probably won't have the time to go over exercises, but if you're interested, you can do it yourself after the conference. Yeah, so next we can plot. Do you want me to make the font larger again? Yeah, so next we can plot some gene expression in space, and these are the marker genes for different types of myofibers. So against the plot spatial feature function, and so here it also shows the that's the log normalized count by default, but you can change the assay. Yeah, so again, that's plotted on top of the myofiber area. So one thing to note here in this function, and actually all the functions in Voyager that where you query genes, it's actually the because ensemble IDs are more disambiguous compared to gene symbols. What? Oh, sure. Wait, how does it show up again?
When you switch tabs, it comes back.
Oh,
Clone does that.
I see. Yeah, so actually, to be more disambiguous, the actual row names of the SFE object used here are actually ensemble IDs, because there are many cases where different ensemble IDs have like the same gene symbol, and so that's why I would use ensemble IDs behind the scene. But if you, however, like the gene symbols are more human readable, and they're so you may want to show the gene symbols on the plot, or you may want to query it on the plot. And if you if you query the data with like a gene symbol, well, it's behind the scenes like ensemble ID, and you find like a multiple ensemble IDs with the same gene symbol, and you'll get a warning. But here that didn't happen. So how to do that is that you specify a this is I'm emulating the scalar argument. You use the swap row names argument to specify like which row data column stores the gene symbols. All right, so that was like some visualizations, and next we can get to some. So we can begin to do some spatial statistics, like Morris I, and before that we can, before we get to like Morris I, we should find the spatial network graphs. So we can do that for myofibers, and here we use the polygon contiguity graph, and yeah, here we also do the. We also find the graph, and this is what the use the plot call graphs function and to plot this graph in space, and this is what the Visium spatial network graph looks like, and yeah, and here. Yeah, and that's what the myofiber spatial network graph looks like. Yeah, so now with the graph, we can get into the spatial statistics, and so there are different ways to categorize the spatial analysis methods. So here they can be categorized with the number of variables. So univariate methods would can only be performed on one gene or one feature at a time. And the bivariate do like 2 features at a time. Like you can look at their like a correlation, but it's spatially informed. The multivariate will simultaneously analyze multiple features at a time, and those can also be categorized as global, which means that they will return one result for the entire data set. So that was just like in bulk RNA-seq, you lose a lot of local heterogeneity. But we also have local results, which would give you a result at each location. So that's a good way for you to explore the local heterogeneity. And also there we also have neighborhood view, which are based on the spatial neighborhood graph. And there's distance view, kind of like Gaussian process models, and using like kernels, just like in the keynote just now. So that's instead of the spatial neighborhood graph, they would directly use the spatial distance. But here in actually I have implemented like variograms. Actually that's distance view, but most of the other methods are neighborhood view. To look at which methods are implemented, you can use the list SFE methods function to see. Yeah, these are the univariate global methods. And to make it quicker, just for this workshop, we can use Scran and Scatter to find the highly variable genes. And they will compute more as I just on those, but they can be computed for like all the genes in the data set. So what is Moran's I? To recap, this is Pearson correlation, and this is Moran's I. You compare these two expressions, and actually Moran's I is kind of similar to a Pearson correlation between value at each location and its and the spatially that version. So it's kind of like a spatially however they're not entirely the same. So like that's so how they're not identical. There's a reference here. So there's like for if you want to run any univariate method on like say something called data, you call data univariate. But actually like there are like these related functions for all the other fields in the SFE object. So if you compute that like a Moran's I for call data like encountered like library size or any genes, and the results are stored in something called like call feature data, and that's how we query the results. So here Moran's I is usually bound between -1 and around 1. So it's just like Pearson correlation. The positive number would mean you have positive spatial hour correlation, and the negative number would indicate negative spatial hour correlation. And here we see that both encountered and genes have like a moderate positive spatial hour correlation. Yeah, and here we do the same for the myofiber area and eccentricity. So you remember that keynote on like the image like cell profiler. So I also trying to do that kind of thing. Yeah, so here instead of like call data univariate, we use anode geometry univariate, and so the result here shows that the area and eccentricity. So area has maybe like a moderate positive spatial hour correlation, while eccentricity is like a maybe weak.
That means that's sporadic, distributed randomly.
Yeah.
Not related with neighbors.
Yeah. If you're like close to 0, then it means like it's more like salt and pepper. They're not related to neighbors.
Okay.
So here we run univariates. The one function rule offer to do any sort of univariate spatial analyses for gene expression. And here we do this for all the highly variable genes found above with Scran. And the results are stored in raw data. So actually, I think Seurat doesn't have raw data. I think, but here I put the raw data field of the SCE object to good use. And here we see the here I'm just showing the which highly variable gene has the largest Moran's I, and some are actually pretty strong. So any questions?
Sorry, what is K?
Oh, it's kurtosis. Kurtosis of the data. Yeah, so here we have some, I put some further reading about like Geary C and more considerations on these. And as just mentioned, global spatial statistics will give you one result for the entire data set. So that means it averages over a lot of local heterogeneity. So we have local spatial statistics, and again we can use the list SFE methods function to find which local spatial methods are implemented in Voyager. So yeah, and our first workshop we will use local Moran's-I. So what is local Moran's-I? So to recap, this is global Moran's-I, and the one below is local Moran's-I. So basically, it's kind of like a local Moran's-I is kind of like a disaggregated and a normalized version of global Moran's-I. Like a global Moran's-I here, you also sum over the i's, but for local Moran's-I, it's I. You do not sum over the i's, but you only sum over the j's, and they are spatially weighted. So yeah, so again, like you use the run univariate function, but type here, well, just now you use type equals Moran, but here you use like type equals local Moran, and that's it. And here we do it just for the Melsted heavy chain too, and just like a plot spatial feature, yeah, here we use plot spatial feature again just to visualize the gene expression again to see what it looks like, and yeah, so image ID, that's how you will specify which image to use when plotting. So analogous to plot spatial feature, we have the plot local result function. So the arguments are mostly the same as plot spatial feature, except that in the second argument you specify which local result to plot, but otherwise it's the same. And here, because we have for local Moran's-I, if there's we have like a meaningful center at 0. So I've used the divergent palette. So the warm color will indicate the local positive spatial autocorrelation, and the cold color will mean will indicate a local negative spatial autocorrelation. So how do you interpret that? So for the warm colors here, those will indicate regions are more homogeneous for this gene. Whereas for these like bluish regions, those are regions that are more heterogeneous for this gene. And if you compare that to the like gene expression itself, then it's kind of like usually regions with a higher expression, like bottom here, they also tend to be more homogeneous for this gene. However, actually, it's not always the case, but often it is the case. Actually, like a local Moran, the function behind it computes many more, so many more aspects. Like they're like p-values, like those kind of things, in addition to the i's themselves. So local result, like so it has a getter and standard similar to like reduced dim and Cauchy geometry. And so we actually have like many other things computed, like the expected value invariance, like a p-value and so on for the local Moran's-I, and the p-value. So this is like already adjusted for the multiple testing. Yeah, this plot shows like how local Moran's-I would relate to the gene expression, and the dashed line show the mean. And so for each local result, for each local spatial method, there's like a default attribute to that plot local result to pool. So but actually you can query like which other attributes are computed for this local result. And so by here in the plot local result, you can change that. So for instance, here we are instead of the II themselves, which so that's the IIs are the default. So here we are going to plot. We're going to plot like the -10 log p-value after correcting for the multiple testing, and yeah, this is what we get. So here, like I use the divergent palette, and the center of the palette is set to like a 0.05, and that's like a standard. So the warm regions will indicate like a significant areas. Yeah, and here, yeah, we can plot like another attribute from the local Moran's-I. So, yeah, so here you see, which regions are like a high value next to other high values, and which regions are like low values next to other low values, and so on. So any questions? Yeah, I think we don't have time to go over the exercise, but we can, actually I also linked to the publications behind some other types of local spatial statistics. All right, so in spatial transcriptomics datasets, we can have like hundreds of genes, like usually for more efficient, to like thousands of genes of interest in like Visium, and nobody wants to plot like localized eye for all of the genes and inspect them one by one. Like nobody wants to do it. So so that's why we have other spatial methods like bivariate and multivariate, and here we can get to a. Here we list the different bivariate spatial methods implemented in Voyager. So those are bivariate globals, just like Pearson correlation. It gives a number for entire data set, and there are also local methods to show like a locally held, say like spatially informed correlation of two genes with varying space. But for the workshop, we use LeastL. So I think Gialdo, like the first version, also implements something like LeastL. Yeah, and here it's that. Yeah, here's the expression. So again, like it somewhat resembles the Pearson correlation. Actually, LeastL comes from the comparison between Pearson correlation and M1SI. But here we also see that it's spatially weighted. So as a result, the interpretation is like suppose you have two genes, and both of them are like a salt and pepper, and both of them don't have spatial autocorrelation, and yet they are strongly correlated in value. So for those genes, for those two genes, because they don't have a spatial structure, you will get a low value of LeastL. However, if you have two genes that are strongly correlated and they are also spatially structured, then you will get a higher value of LeastL. So again, like so here we still use like highly variable genes, and so actually for LeastL, the original implementation in SPDEV is pretty slow, so I wrote my own implementation. Now here I used calculate bivariate instead of run bivariate because because the global bivariate methods outputs have very different formats, and there's no obvious way for me to store them in the SFE object. So there's no easy way for me to like put in like row data or something. So actually for univariate and multivariate, they're also like calculate univariate and calculate multivariate functions. So that's to emulate the scalar function, which has like a run PCA, which will store the results of PCA in the SE object. And there's also calculate PCA, which would not store it, but just returns the output directly. And here, that's just to line 680. That's just to get a divergent palette. And here we plot the heat map to show the correlation matrix, which is also spatially informed. So potentially this can be used for like a G-code expression analysis, like a WGCNA. I think it will be interesting to see, but I haven't tried it yet.
I have a question.
Yes.
So there is a correlation within each cell. What is this correlation?
So it's a.
You have two genes, so that's correlation between those two genes inside each cell. How about the spatial?
Well, so again, like when you look at the equation, like the Wij's are the spatial weights.
So how to calculate that spatial weight? Okay.
Yeah.
You
have the graph, and from the graph you can compute like different kinds of edge weights.
Kernels like this. Okay.
Yeah, actually, for this one's not kernel. It's a spatial network graph. So just now, like when I was talking about like call graphs, there are different ways to find this graph, and also like different ways to compute the edge weights.
Okay, so just can be calculated.
Yeah. All right, maybe I'll, for interest of time, if you're curious, you can read these explanations. But All right, so next we can get to the multivariate methods. So here, with list SFE methods, you can see like which multivariate methods are implemented in Voyager. Actually, although like Voyager already has many ESDA methods, you can the user interface is designed to be extensible, and there's a vignette just about that. So which is here. Yeah, so this vignette will show you how to extend Voyager. It's just like tidy models. Yeah, it's really inspired by tidy models. So you can this way if you have your own new methods, and you can this is how you get Voyager's uniform user interface to run your new methods and to get it to store the results in the SFE object. So as a reference, so here we're going to do the MULTISPATI PCA, which is a type of spatially informed PCA, and it was originally from ecology. Yeah, so it comes from ecology, and it's although like it has an original implementation in ADE spatial, which is pretty slow. So I re-implemented that with our spectra, so that way it can scale to like hundreds of thousands of cells. Like I ran all the analysis on my laptop and it was fine. So here we can use a larger number of highly variable genes, and so that's I'm just using Scalar to run PCA. That's just standard good old non-spatial PCA on this data set.
So which bit is that the BS param which is doing?
Oh, so that's in Bioconductor. There's a package called BioC Singular, which has different algorithms for like SVD. So BS param is to specify which algorithm to use.
So this is just doing normal PCA.
Yeah, that's non-spatial PCA.
Okay.
That's in Scalar, it's not in Voyager. Yeah, here with elbow plot. Okay, this is a Voyager function. It emulates the Seurat one because Scalar doesn't implement a nice function to plot the eigenvalues. Yeah, then we can plot the gene loadings for the PCs. So this way you can see you can spot some marker genes, and yeah, for PC1 you see like here like the mitochondria genes, which makes sense because it's the skeletal muscle and they have a lot of mitochondria. Where's those above? I think actually these also these are in the these genes on top are in the inflammatory the injury site, and I think FTL1 it makes sense because macrophages play an important role in iron metabolism. And here I have a spatial reduced dim function to show like the spot embeddings in each PC in space, and again I use a divergent palette to because like PCAs can get like positive and negative results. And for non-spatial PCA, so because cell types are spatially localized, so for first two PCs, they actually are spatially, actually first three, they are spatially structured. The first one was separated most of myofibers from the injury site, and the PC2 is also kind of about the injury site, and the PC3 is kind of about the muscle tendon junctions. But PC4 looks like it just got an outlier. And here we run the MULTISPATI PCA to see how the results compare. So basically, it's basically you add like that basically you put the spatial neighborhood graph, the adjacency matrix with the spatial weights. You add that to the when you're computing the covariance matrix. For in standard PCA, you would diagonalize the covariance matrix, but here you in MULTISPATI PCA you would diagonalize the spatially weighted covariance matrix. And actually, the interpretations of the eigenvalues are very interesting. In standard PCA, the eigenvalues are the variance explained by each PC, but for MULTISPATI PCA, the eigenvalues are the product of one's I of the cell embedding in that PC, and and the variance explained. So in order for you to get a highly positive eigenvalue, you not only have to explain more variance, but you also have to have like a high positive variance I. But meanwhile, like you can this spatially weighted covariance matrix is not positive definite, so you can get a negative eigenvalues. So to get negative eigenvalues, like a very negative one, you not only have to have negative spatial autocorrelation, but you also have to explain more variance. So for Visium here, we do see like there's like a very positive one, just like an non-spatial PCA. But we don't have like many significant, like we don't have like a very negative ones. However, like in if you go to the documentation website of Voyager, there is like an example of negative. Yeah, like in Murfish, there is an example of negative spatial autocorrelation, like here. Yeah, so this is the this is for the Murfish liver data set. You see there's like a very negative one. So it turns out like from
So in your example for the vignette that you're running through, is it possible because you haven't calculated enough PCs to see that? Because in this one, if you truncated it like in your other vignette, if you truncated like 38, you would have not seen the.
Oh, actually, it's sorted.
Down jump.
I have only computed like the 20 most positive and 20 most negative, so it's already sorted.
Okay.
Oh, okay.
Yeah, so actually this one's already sorted, but for Murfish, it's at a single cell resolution. We like at least in the liver, I haven't looked at other tissue, but in the liver we do see like a very negative one. And actually, based on this is the most negative one, and based on the gene loadings, it's actually it separates the hepatocytes from the Cooper cells and the endothelial cells. Yeah, it can be pretty interesting. I think it's just like in, well, I said like from geospatial to spatial omics. In geographical space, it's just like the 15-minute city and as opposed to like suburbs and zoning. So you have to drive far away to go shopping if you only have positive spatial autocorrelation. So in contrast, if you have a 15-minute city, like your shops, like your grocery and so on, like your jobs are closer to home. So in some cases, like functionally, negative spatial autocorrelation would make sense. But here for Visium, because in Visium, it doesn't have single cell resolution, you're actually binning over many cells in space. So the interpretation of like Moran's I or any spatial metric will be different from those from Murfish, which has single cell resolution. And I think that probably because of the binning, it smoothes over a lot of the negative, like what may otherwise be a negative spatial autocorrelation. So we don't see it here. Yeah, so here we can plot the gene loadings. So those are the contributions of each gene to each PC, and this is what it looks like in space. So PC1, PC2, they kind of look like in the standard PCA, and PC3 maybe still, but notice here like PC4 is also very spatially coherent in contrast to standard PCA, which I can still pull up here. Yeah, in standard PCA it's like this. But for like MULTISPATI PCA it's yeah, it's like this. So maybe because the PCs themselves, like when you project the Visium spots in them, they tend to be more spatially coherent. So you may wonder like if like in standard single cell experiment, like single cell RNA-seq data analysis, like we will like first do PCA and then you find like a k-nearest neighbor graph in PCA space, and then you do like a latent or low vein clustering with that graph. So this is what I do here, and I do the same in MULTISPATI. So actually, at least for this data set, because the PCs for MULTISPATI they're more spatially structured, I do get more spatially structured clusters. Yeah, and on the left is from the non-spatial PCA, on the right is for the MULTISPATI PCA. However, this I have it's just kind of an interesting ESDA experiment. I haven't benchmarked this compared to like base space and so on. I think like base space might do better. Although like this is way faster than like base space. Actually, we can. So here, why do we get like a more spatially structured PCA? Actually, with Voyager, we can also run like Morris I on the dimension reduction cell embeddings. Like here, I'll do it for like the cell embedding in PCA and MULTISPATI PCA. Here, yeah, it's just some like tidyverse to. Yeah, just like some tidyverse thing to plot the Morris I for each PC. And here for the sake of plotting, I also compute the actually there are theoretical bounds of Morris I given your spatial neighborhood graph. Actually, they're not exactly -1 and 1 as in Pearson correlation, and so usually this the bounds of Morris I are like for the upper bound is like around 1 usually, just like Pearson correlation, but lower bounds usually like around -0.6 instead of -1. So yeah, I'm computing the Morris bounds to help me to interpret the negative Morris I that will show up here. Yeah, the upper one, the upper curve shows the Morris I for each principal component from MULTISPATI PCA, and the one in the bottom are the negative ones. Actually, like there are like many combinations of genes, like linear combinations of genes in this data set that have very negative spatial autocorrelation, considering the lower bound of Morris I. However, they explain very little variance, so maybe we shouldn't really overinterpret those. They might just be noise. In contrast, here in the middle here, the curve is the Morris I of the Visium spatial embedding in each PC for a non-spatial PCA. So see the Morris I decline drastically from PC1 to like PC5, and then it bounces around there. So yeah, because of higher Morris I for the positive ones, we get like a more spatially coherent clustering. However, like in other data sets, when this Morris I for MULTISPATI also decays like in non-spatial PCA, then you will not get very spatially coherent clustering. So that's it. Thanks for coming. So finally, like Voyager is not perfect. It's not really meant to be the most comprehensive ESDA framework in the world. It's meant to complement many existing methods like Bayes space, like Lisa Clust, which is also like a statue from Alice Patrick's lab, and so on. So there are also other caveats like Geodo and SquidPy implement some ESDA methods for categorical variables. However, that's not done in Voyager yet. I think I would actually, it's really easy to do. Like SPDEF already has the joint count thing, and it's really easy to add it. But meanwhile, I think there isn't like a perfect agreement. I don't think there is like an agreement on what exactly is a cell type. I think I would need to have more considerations on like what really is cell type before I also like more considerations on categorical spatial analysis before I dig in further about that. I think also so far these methods are run on each section or each sample or image separately. Actually, for multivariate ones, you can run them jointly, but actually, I think this is a unique aspect of spatial omics data, because in geospatial data you can't get like a technical or biological replica, because each city is very unique. They have their unique histories. They're not replica. However, in biology, we often have a case and control, and for each group we have multiple subjects, and like also make multiple samples from the same subject and so on. I think more actually I'm currently looking more into that when I actually do have a data set with case and control, and yeah, so with case and control and multiple samples, I think there will also come like a problem of the manifestation of batch effect in the spatial aspect of the data. I think it could happen. Yeah, I think that's it. So thanks for coming. So do you have questions?
Thank you very much. Wonderful workshop. No question, yes.
Wow, fantastic work. That's a
Alright,
real
thanks.
tour de force. Have you considered contributing any of your speed improvements that you've made with some of these like type functions that you had from like SF, where you said you'd re-implement it?
So you mean
Back-
contributing? You mean like contributing my speed improvements to like SPDEP?
Yeah, back up.
Yeah, I actually thought about that. I sort of like I don't quite have the audacity to message them yet, but I think I should open the issue. Yes.
I've always had these things about R packages. And it looks like you could do this, but this kind of R markdown methodology, you could have like a check suite, make sure it's working okay, kind of suite of R markdowns. You just run them and make sure everything's running.
Yeah.
So if you have a different version, you just run them automatically, or you think?
Yeah. So do you mean the documentation website?
I mean, just
Bioconductor does that by default.
I'm sorry.
Bioconductor does that.
Oh, do they?
Yeah. So Bioconductor. Yeah. So actually, like for my website, it's not built on Bioconductor because Bioconductor has a 5 megabyte size limit for install packages, and actually all these plots like they add up. Like the website's kind of like a book. It's like way larger. So for Bioconductor, I have a separate small vignette, but this one's like it's in the same GitHub repo, but it's in a separate branch from main.
Yeah.
So it's synced with main branch by merging from the main branch. So like this is the main branch. I have a smaller vignette, and the website's actually in the documentation branch.
Yeah. I mean, there's different kind of sanity checks people can run on
Yeah.
packages, whatever.
So,
but if you had like a major version change, you could kind of have a regression that would compare it, and it wouldn't give you a book, but it would give you a guide to say how does it match what you previously did, or is it?
Yeah, like
Is there?
yeah, I have unit tests. I also have unit tests for the plumbing function, so that way if it breaks, I'll know. And so after Bioconductor runs like RCommand check and BioC check every day, so those RCommand check would run all the unit tests. So if it fails, Bioconductor will send me a notification. Also for a documentation website, it's so actually we have like a develop version of the website which we mess around, and then that's automatically built every day. So if it breaks, I'll know.
The reason I asked because there's a project that was a while ago for validate R or validated R for clinical work, and a lot of the packages in CRAN they don't really have detailed regression tests. They have basics. But I like the fact that Bioconductor has this in there built in because it really helps out.
I would say it's maybe not built in per se. I think CRAN doesn't require unit tests per se. For you can pass RCommand check without having unit tests, but Bioconductor does require you to have unit tests.
Yeah.
So actually, I try to improve my code coverage. Right now, like 80-something%, but I try to make it better so that way if I have regression, I would know. Yeah. And the unit tests are run every day.
Right.
And just a clarification, our validation was particularly for pharma and biotech companies that were looking to identify certain Bioconductor packages to work in the context of validated systems for submission to FDA and your EMA. So, but the Bioconductor, every Bioconductor packages, and like I said, is you've got to pass it. It's unit tested.
Yeah.
State, or you start getting spammed from bias.
Yeah. Yeah, honestly, like I feel like there should be a Bioconductor. I think it should have stricter standards for unit tests.
With 3,500 packages, there's always this tension between trying to improve the quality and trying to keep all the volunteers that have contributed to the work with us. So we are. That's not about. That's actually not a topic about. I don't want to steal your time. That's a topic. It's a more that's a both. It's really interesting. Computation biology.
So that's it.
Well, thank you, Kayla.
Yeah, also like I should, yeah, it's on the slides, but I got to the website, so I think I should thank other members of the Voyager team and also the Icelandic colleagues who did the Python implementation, and also everyone in the lab and the funding from NIH and the Icelandic Research Fund.
Perfect. Now it's a perfect time.
All right, thanks for coming.